So welcome back to the session on proteins, and here we'll be discussing on classification of proteins. Uh, this is actually a more comprehensive and a popular classification of proteins, widely accepted one, and this is based upon the amino acid composition, the structure, shape, and solubility protein properties of the proteins. And based on that, proteins can be divided into three, that is simple proteins, conjugate proteins, and derived proteins. Okay, what are these? Simple proteins, they are composed only of amino acids residue. So when the simple proteins hydrolyzes, it produces only amino acids. Conjugated proteins, on the other hand, they have proteins plus non-protein moiety. Okay, so amino acid part plus the non-protein part together form the conjugated protein, and these non-protein part is known as prosthetic group, which will be learned in the next slide. We will be looking into derived proteins. On the other hand, you can see here, these are actually the degraded products of simple and conjugated protein. So when simple and conjugated protein gets degraded or broken down, denatured, what we get, those are included under derived proteins. I hope it is clear. Okay, simple proteins, it is further divided into fibrous and globular. The fibrous protein, it is as the name suggests, it is fiber like. Globular means spherical, globe like, isn't it? Spherical or oval in shape. So based upon the shape, the simple proteins are divided into two, fibrous and globular. Obviously, because of this nature, there are different, they show different physical properties that we'll be looking in detail in the coming slides. Okay. Conjugated proteins, on the other hand, they are divided into various groups based upon the prosthetic group. As already mentioned, conjugated proteins are the proteins which contain protein or amino acid moiety as well as amino acids plus what you call non-protein moiety. So when conjugated proteins hydrolyzes, what all things are produced? It will be amino acids plus non amino acid or non-protein part as well. This non-protein part is what is referred as a prosthetic group. So based upon the prosthetic group, we can have different kinds of conjugated proteins like nucleoproteins, mucoproteins, glycoproteins, metalloproteins, lipoproteins, phosphoproteins, etc. There are plenty other. Okay, so I hope the classification is clear. Okay, we'll look in detail. Simple proteins, as already mentioned, it contain only amino acid residues and no other chemical constants. So when it is broken down, when it is hydrolyzed, we get only amino acids. Okay, example is ribonuclease A and chymotrypsin. The major examples, major types of simple proteins are globular protein. Globular protein, they are spherical in shape, spherical or oval in shape. They are soluble in water or other solvents, and they are digestible. Okay, it includes albumins, globulins, and histones, which you are familiar with. Albumins, it is soluble in water. Okay, and it is present in blood. Serum albumin is there. In egg, albumin is there, and in milk, lactalbumin is there. So wherever it is situated, albumin is a water soluble protein, globular in nature. Okay, globulin, as the name indicates, in the name itself, we have the term, isn't it? Globulin, right? Serum globulins are there. That is just like albumin. In the blood, we can find the globulin, right? And what about histone? Histone is a protein. It is a strongly basic protein, actually, which is found in the uh, the chromosome structure formation. Okay, like you will be learning it in your molecular biology classes, how the histones come into play for the formation of chromosomes. Okay, so the all these are globular proteins. What about fibrous proteins? Fibrous proteins, they are fiber shaped, insoluble in water, and resistant to digestion, so they cannot be easily digestible. They are not easily digestible. Okay, and examples include collagens and keratins. Collagens, they are connective tissue present, actually. I hope you know that they are found in the connective tissues, like the in the matrix of the connective tissues. Okay, uh, collagens on boiling with water or dilute acids, they yield gelatin, which is soluble and digestible. Okay, so gelatin, which are used in the manufacture of jelly or ice creams and all, it is produced from collagens. Once it is boiled, it produces the soluble, digestible form, that is the gelatin. Now, keratins, they are present in the exoskeletal structures like hair, nail, horns, etc. And obviously, you know that these are indigestible, and obviously insoluble in water. Okay, so this is regarding the simple proteins. When you go for the conjugated proteins contain non-protein part as already mentioned, and these non-protein part or non-amino acid part, it is referred as the prosthetic group. Okay, and based upon the prosthetic group we can see different groups. Okay, like lipoproteins, glycoproteins, metalloproteins. The name suggests along with proteins they have lipids along with it. Okay, so when it is broken down we get proteins as well as the lipid molecules. Then glycoproteins, it contains sugar along with the protein molecule. Metalloproteins, it contains specific metals. Okay, metals in the sense it can be iron, copper, uh, magnesium, zinc, etc. Okay, so nucleoproteins. So here the nucleoproteins, the nucleic acid is the prosthetic group. For example, nucleohistones, nucleoprotamines, they are examples. Okay, glycoproteins, the prosthetic group is carbohydrate, and examples it is mucin, ovomucoid, etc. Lipoproteins, they are proteins found in combination with lipids as the prosthetic group. It includes like membrane lipoproteins, serum lipoproteins, etc. Phosphoproteins, it contains phosphoric acid as a prosthetic group, and it is present in the casein, the protein in the milk, and vitelline, the protein in the egg yolk. Okay, then we have is the metalloprotein. As already mentioned, it contains metals, metal ion in ionic form. Okay, metal ions like iron, copper, cobalt, zinc, magnesium, etc. Zinc is a prosthetic group in carbonic anhydrase. Okay, the enzyme which is involved in respiration, carbonic anhydrase. It is zinc is the what you call metal ion present as a prosthetic group. So these are the different conjugated proteins. As already mentioned, derived proteins include those products which are formed by the hydrolysis or breakdown or denaturation of the simple and the conjugated proteins, and it includes coagulated proteins, proteins, then polypeptides, peptones, peptides, etc. Okay, so this is the classification of proteins, and next we pass on to, yeah, here it is just a table which shows the different kinds of conjugated proteins with the prosthetic groups. Okay, you can see here, right? Zinc, alcohol dehydrogenase, iron, ferritin. Ferritin is a like a protein which is involved in the storage of this one iron. Actually, it is a storage protein. Okay, then calcium in calcium calmodulin complex. It is involved in many processes in what you call the muscle contraction and all. Okay, so here I hope this is clear with you. Okay. Next, we will be passing on to the analytical techniques, which we'll be learning. The first one is the Biuret test. In the Biuret test is actually a colored reaction, which is also considered to be the one of the important properties of shown by the proteins. Okay, here what we do is we take the protein solution, add an equal volume of sodium hydroxide solution, and mix it thoroughly, and then we add copper sulfate solution drop by drop. Mixing between like drops until a what you call purplish violet color is obtained. Okay, so when we add this one copper sulfate solution, what happens is purplish color develops. Okay, and till it sustains, we have a sustained purplish color, we can add the copper sulfate. Okay, so the Biuret reaction is given by substances which contain the CONH2 group. Okay, that is amide, and join together directly or through a single atom of nitrogen or carbon. So positive reaction indicate that the given solution, given protein solution contains at least 2 peptide bonds. So it has to be a tripeptide. Amino acid doesn't give Biuret test. It is given only by a protein or amino acid chain with at least 3 amino acids. Okay. Now, what is the chemistry? What is the principle behind it? The molecule, the proteins, it should be having a CONH2 group, then only it can develop that color. And the violet color is due to the formation of a copper coordination complex. Okay, so you may have learned in coordinate chemistry the coordination complexes which can be developed by the copper compounds, right? And this is what happens when CuONH2 is being acted upon by the complexes in the Biuret reagent. Okay, so this is regarding the Biuret test. Fine. Okay, so we have been speaking on the Biuret test, and this is how the test will be. So when we add, you can see sodium hydroxide solution, copper sulfate solution, right? Or Biuret reagent, we can see. So when it is added, this is how it will be. Okay, only when there is a protein in that, what we said is actually tripeptide, isn't it? So at least two peptide bonds have to be there to give such a purplish color. Okay, it is not a precipitate, it is a purplish color. Okay, and this is a positive result. Okay, so once we get this positive result, we can say that it contains protein. Okay, amino acid chain. Now the next test, it is the Xanthoproteic test. Xanthoproteic test here, 1 ml of concentrated nitric acid is taken, and then test solution is like uh, mixed with it, and what happens as a result is a white precipitate is formed on adding nitric acid, okay, and which on heating turns yellow and then dissolves to impart yellow color to the whole solution. So what happens is actually white precipitate is developed, which on heating it becomes you can see it becomes slightly yellow in color, right? And slowly the whole solution becomes completely yellow. Okay, but upon adding alkali, the color deepens to attain orange color. Okay, so we can see the change from when what happens in an acidic medium to what happens in an alkaline medium. And what is the chemistry? Here, addition of nitric acid causes denaturation of proteins. So proteins, one of the very important what you call denaturation agent is an acid. Okay, so when nitric acid is added, proteins get denatured and it becomes white precipitate. Okay, uh, uh, and yellow color develops due to nitration of benzene ring of amino acids tryptophan and tyrosine. So what does it say? That is tryptophan and tyrosine. If it is present in the protein, it gives a positive result for the Xanthoproteic test. Okay, so it is the tryptophan tyrosine which contains a benzene ring, right? It is actually aromatic in nature. So this on nitration gives a yellow color. On addition of alkali, it increases the ionization of compounds, and hence the color deepens and becomes orange color. Okay, so this is the color, orange color, which develops when the alkali is added after the what you call acid treatment. Okay, this is the last one, Ninhydrin test, which we have already mentioned, but we'll see how it will be. So this is how the Ninhydrin gives coloration when it is added to any amino containing compounds. Okay, it develops a blue purple color. In the case of proline, what happens? Such a kind of what you call complex cannot be formed, and hence it becomes yellow orange product. Okay. Otherwise, for all the amino acids, it gives such a blue complex, blue purple complex, and this is due to the, we have already mentioned, it is because of the complex formed, right? Which contains Ruhmann's purple, that is the color actually. It is because of the ninhydrin and ammonia complex. Okay, and this gives a faint blue color. Okay, fine. So this is it. Thank you.